All right, thank you so much for inviting me. And I'll keep this really, really basic at the request of Ron and Shamir. So basically what I'm going to cover is what are regulatory elements, how we look for them, functional assays for them, and biochemical assays for them. And I'll end in the end with one example from our lab. But as Ron requested, I'll keep it very general. And I think later on in the assignments, when there's gene regulatory workshops, I'll talk more about the work that we actually do. This is, again, very basic, and just to make sure that everybody's online and same page to get to this. Okay, so as geneticists, we're really, really good at finding genes. So if this sequence, for example, was a gene, we'd probably pick it up because genes have these rules that we know. They start with an ATG, they end with a TGA. They have exons and introns that start with AG, GT. So we get sort of an A for finding genes. We're good at this. But if this sequence was a regulatory element, it would be hard for me to figure that out. And one of the main reasons is that we're faced with the problem that 98% of our genome actually doesn't code for a protein. And so we have this vast genomic space to search for regulatory elements. In addition, for genes, we have this code that we know. For example, CCC codes for proline. And we also know the consequences of nucleotide changes to that code. You change CCC to CTC, proline changes to leucine, and that could lead to some sort of phenotype. But we don't have all that for regulatory elements. So the problem is, even if I find a regulatory element, I don't know the code of it. I don't know what it's going to be doing at this stage. And if I find a mutation in it, a nucleotide change in it, I really don't know what it's doing on a phenotypic level still at this stage. And a lot of the work that my lab has been doing, which I'll show later on, not today, is really trying to get a better understanding of this code and what mutations in these regulatory elements could lead to in terms of phenotypes. And as you'll see, there's still a long way to go with that. And we've also been working on developing a lot of high-throughput technologies to allow us to get a better sense of this. So why do we care about these regulatory elements? Well, if you have nucleotide changes in these switches that tell the genes when to turn on, when to turn off, that could lead to differences in gene expression. And those differences could lead to phenotypes like human variation between people, morphological differences between species. It's thought that a lot of differences between species, for example, if you compare humans to chimps, there's not a lot of differences in protein coding sequences, but the main difference is probably due to regulatory differences. And so finding these switches and the changes in them can lead us to better understanding of this happens. And also human disease. And there's now more and more cases that are coming out showing that a lot of mutations in these regulatory elements can lead to human disease. And for example, if you look at all the genome-wide association studies that have been done, GWAS, on humans for a variety of diseases and other phenotypes like pharmacogenomics, the majority of them, almost 90%, actually hit non-coding regions and do not hit genes. And so clearly there's a lot of human disease that could be caused by mutations in regulatory elements and not in genes. So as I mentioned, what we're going to do today is talk about the different kinds of gene regulatory elements. How can we find them? How far away to look for them? Functional assays that we and others do to test their function. Later on, a bit of regulation in 3 dimensions, 3D, and then example from our lab. And this is completely informal, so really at any point, if you have a question, please don't hesitate to ask, okay? All right, so the different kinds of gene regulatory elements. So promoters, I'm assuming everybody knows promoters. Is there anybody? Okay. So here we have a gene A, let's say, and it has a transcription start site right here, and what's called a promoter that basically turns on gene A. And here I'm showing you a mouse embryo that was stained with LAC-Z for the expression of a gene. So this gene is expressed here in the limbs, in the developing neural tube, and in the brain here. And this promoter, you would think, could drive this expression pattern. However, what people have seen is that if you inject just the promoter and this gene, you don't get this complex expression pattern in most cases. However, if you inject a bigger chunk of DNA, you start seeing this complex expression pattern. And the reason is that they are, well, so before I jump to that, I forgot to mention, So in terms of promoters also, one thing to keep in mind that there could be alternative promoters. So you may have promoter A and promoter B for the same gene, and that could lead to differences also in splice variants and differences in expression. So that's an important point that I forgot to mention. And going back to promoters not being sufficient, so as I mentioned, promoters can sometimes not be sufficient, and if you inject a bigger chunk of DNA, you start getting this expression pattern, and that's because there are sequences called enhancers that tell the promoter to turn on, for example, here in the limb, here in the neural tube, and here in the brain. So the enhancers are sort of the promoters of the promoter, and they basically tell this promoter to light up in this tissue, and combine these enhancers and the promoter give you this complex expression pattern. There are also other regulatory elements. Probably the least studied ones are called silencers. So silencers are basically the opposite of the promoter, and they can basically silence the promoter and shut down this gene by either looping or other ways, histone modifications and so forth. And one of the reasons they're not as silenced is, as I'll show you later, there's not really good functional assays yet to find silencers. There's also, especially in my lab, a big argument whether silencers are just enhancers that are silenced or do silencers exist, but that's another thing. Insulators is another group of regulatory elements. So let's say we have a gene here called gene B, and it has an expression right here in the eye and has a promoter. What could happen is that these enhancers, for example, this limb enhancer, could start turning on this promoter right here in the limb. And now suddenly you have a gene that shouldn't be expressed in the limb being expressed in the limb. And so there are DNA sequences called insulators that are here, which you can imagine them as being like a Chinese wall, and then the China wall. And then they basically insulate and don't let this sequence go here and turn on this promoter. It can only go here and turn on that promoter, in a sense. That's very sort of simplifying it, but that's sort of the idea of insulators. Are all of these clear for everybody? Since we're going to talk about, yeah.
Probably stupid question.
No such thing.
So I've seen, are there assays to identify promoters, or do people just define it as like within a certain region of the transcription start site?
No, so definitely there are assays, and I'll show you later on how people basically find promoters. It's quite simple, where you put a sequence that you think is a promoter in front of a reporter gene, and you see if it lights up and where does it light up. So yeah, definitely, and a lot of people do that. Yeah.
Also, a simple question. So, is it true that you don't necessarily need to have a linear order of things? Like, can enhancers be located somewhere else? Would you know?
Definitely. So, great question. So, yeah, enhancers can be located 5'. They can be 3' to the gene. They can be even on another chromosome. So, there's a nice example for the H enhancer, which is an olfactory receptor enhancer that's actually on another chromosome, binds to another chromosome, and enhances. So, yeah, they could definitely be anywhere. And in terms of how far away to look for enhancers in cis, we'll talk a little bit more later on also. Any other questions before I continue? All right, so this is clear since I'm going to talk a lot about these regulatory elements, in particular about enhancers. Okay, so these are sort of the different flavors of regulatory elements. I guess going back, I think we still really don't have a great understanding, and I think there's a lot of different regulatory elements. I think there's a lot of different types of enhancers, and I think there, hopefully the field will move forward in the years to come. But I think, enhancer is a very big general term, sort of like a car, but there are very different kinds of cars. And so I think there's a lot more work to do to define enhancers and also these other regulatory elements, and hopefully we'll see a lot of that in the future. But for now, I'm just using a general term: enhancer, silencer, insulator, promoter. So how can we find these regulatory elements, and what can we do to find them? And so there's quite a lot of different ways to find them, and I'll talk just about a few of them, sort of the more common ones. One, it's thought that transcription factors bind to these regulatory elements and activate them or silence them and so forth. So you can look just on the sequence level at transcription factor binding sites, which we'll talk about. Another is comparative genomics, comparing genomes in evolution. Another is CHIP, and there are other technologies, but CHIP is probably the most commonly used, so I'll talk a bit about CHIP, and then also DNA-seq and ATAC-seq, which is now quite widely used also. So we'll start with transcription factor binding sites. This is pretty simplified, and the talk after me will be a lot about transcription factor binding sites and motifs, so I'm really going to keep it quite simple. So here we have a gene, here let's say we have an enhancer, and here this alien spaceship is a transcription factor that basically binds to this sequence. And so if we scan, for example, the genome for this sequence, we can try and find that we know this transcription factor likes to bind to. We can find regions in the genome that could be regulatory elements. However, it's not that simple, as you'll probably see also in the talk after me, because sometimes this transcription factor likes to bind also to this sequence, which is a bit different, but has an A here, or this sequence, which is a bit different. And so what people have done from transcription factor binding sites is developed what's called position weight metrics, which again, I'm sure you all know, but just in case. So if we look at the position weight metrics, for example, for this position, most of the time you see an A, so let's say 60%, and 40% of the time you see a T in this position that the transcription factor likes to bind to. Second position, TTAA, so you see 80%, 20%, and so on and so forth. So these are called position weight metrics, where you can sort of give a transcription factor binding site estimate of where it will bind and how it will bind at what percentage. Is this clear for everybody? Okay. Another way to look at these is logo plots, which is a very nice graphical way where basically the size of the letter gives you the size of the binding. So for example, here you have an A that's much higher than a T because 60% of the time you get a T. Here 80% of the time, 60% of the time you get an A, and here 80% of the time you get a T, so you would get a very large T versus a very small A, and so forth. So these are the logo plots, which would be a similar way to look at this. Okay? So there's a lot of transcription factor binding site programs. This is just a few. This is a bit old that you can use to find these, and I won't bore you too much. This will be in the slides, and again, the next talk will be a lot on motif searches. And so one way you can think about is you look at the sequence, you look for transcription factor binding sites, and you can speculate that a TF might bind there. However, the problem is that, for example, for a 6 base pair sequence, there could be a lot of places to look for, and so you really need to have improved ways to finding these, and I'll show you one example of how people improve this. So another way is to find these regulatory elements by using comparative genomics, and that's been quite used extensively until about, say, 5-7 years ago, before CHIP really kicked in, ChIP-seq, which we'll talk about later on, but it's still being used and still a nice way to find potential regulatory elements. And so the idea here is that you just take advantage of evolution. So for example, here, if we compare a non-coding sequence, not a gene, between human, mouse, chicken, and fish, we can look for regions that have been conserved throughout evolution. For example, this region right here. And if it's conserved, you think that it might have some kind of function, and that function could be being a regulatory element, either enhancer, promoter, insulator, silencer, or so forth. So you can take advantage of comparative genomics to try and find regions that could be functional in the genome. And this is another website that is quite easy to use that I recommend if you want to do comparative genomics, the ECR browser. And there's also the UCSC genome browser that allows you to use this. Also, the ECR browser is really web-based and very easy to use. What it does here, for example, is it's comparing human to fish, frog, chicken, opossum, mouse, and dog. Each one of these tracks basically is a comparison to one of them. And if you get a peak, that means that that sequence, here's an exon, for example. You can see that that sequence is conserved all the way from humans to fish in this position. And that's usually a lot of how comparative genomic tracks are used. Also in the UCSC genome browser and others is basically giving you a peak of conservation, show you that it's conserved. Going back to the transcription factor binding problem, as I said, 6 base pairs can be quite common. In the human genome, for example, 734,000 times. And so how do you make sense of what could be functional and what could be not? So one way is to use conservation that I just showed you. For example, if we compare human to mouse, and we have that same sequence here and here, this one is based on conservation. Since it's not conserved, you can speculate it's less likely to be a functional transcription factor, well, this one is more likely to be a functional transcription factor. So using comparative genomics, in a sense, can allow you also to take motifs and think which ones are more functional and which aren't. And of course, there's many other ways to sort of filter and screen through motifs and so forth. Clear so far? Okay, so one nice way, and that's another reason for showing this program, UCR browser, to do this is in any one of these peaks, if you click on the peak here, that will open up the window here that shows you the conservation and the comparison, in this case, between frog and human. So you can see that this sequence is conserved over 225 base pairs, 72%. And another nice thing is when you click here on conserved transcription factors, it takes you to a program called RVista that looks at transcription factor binding sites in combination with conservation. So for example, you can see that this sequence is conserved, and here you have a lot of transcription factor binding sites in the conserved sequence. And so you would speculate that they're probably functional, and that will help you sort of narrow down the regions of importance and the regions of function where that is. Clear so far? Okay, so comparative genomics can also be used just to annotate new genomes and so forth, and this is sort of a joke I always show. So let's say I walked down in Berkeley and I found this alien here that is very green, and basically I want to figure out why it's green. I can, grind it up and make DNA, and then I sequence it and I find that the alien DNA matches jellyfish GFP, and so using comparative genomics can also really assign other functions like protein function and also other things. And so it's quite a useful tool also nowadays where a lot of people are using ChIP-seq and other biochemical technologies, I think, to find function and to find a better understanding of what things are doing, both regulatory and also coding. So comparative genomics, as I mentioned, is probably the technique that's most highly used now for gene regulatory finding. And the reason that is this is a sort of a nice experiment where people looked at how they could predict enhancers based on various techniques. So if we look at comparative genomics, you have a 20 to 50% chance of finding something that's an enhancer. If you add to it transcription factor binding sites, like I showed you before, it gives you about a 50 to 60% chance, so it's a good increase. But for example, if you use ChIP-seq for P300, which we'll talk about in a minute what it is, it's a good mark for enhancers, you really increase your success rate in finding functional enhancers to 87%, as you can see here. And same in terms of finding tissue specificity. If you want to know if that enhancer, for example, is an enhancer in the brain, if you use comparative genomics, that's going to be hard to do. Transcription factor binding sites might help you because you may know that there are some transcription factors that are brain specific that bind in the brain and activate things in the brain, so that could increase it to 50%. But if you use ChIP-seq, 84%. So again, this clearly sort of shows the advantage of ChIP-seq in being much more accurate in finding regulatory elements. And this is, I think, a bit of an inflation. I'd say it's probably a bit lower than this in actually finding functional sequences, but still does much, much better than these guys, which are also a bit inflated, I'd say, in this sort of example here.
But you're talking now what fraction is a true positive, right? So you miss still a lot when you do that, right?
Yeah. So in all of these, I think when you test them, a lot of them are not enhancers in the sense. So you have a lot of false positives. That being said, it also depends on when and how you test it. And yeah, so in this article that I'm sort of citing that I'll show later on, they had a great success rate, but I know how random their choice was. And usually as a biologist, you choose near genes that you're very interested in that have an important function in there if you choose. So, but what I do agree with this a lot is it definitely gives you a better success rate than these techniques. And I think combining all of them, and there's now a lot of tools like Enhancer Finder from Katie Puller's lab and others, that if you combine all of these things together with a lot of other things, I think can really improve both your ChIP-seq peaks, predictions, and so forth. But you still have a lot of work to do, I completely agree. So what is ChIP-seq? So ChIP-seq basically, what you do is you take your tissue or your cell line of interest and you use formaldehyde. And what formaldehyde does is basically makes the DNA binding proteins, the transcription factors or anything that binds to DNA, gets stuck on the DNA. So now you have basically DNA with everything that binds to it sort of stuck on the DNA. And then you chop the DNA to smaller pieces so you can later on be able to sequence it. And so now you have small pieces of DNA with, for example, this red transcription factor bound to the DNA, or these blue transcription factors bound to the DNA. And then the trick is you use an antibody that is specific to that transcription factor that you're interested in. So let's say I was interested in this red transcription factor. I use an antibody for it, and that way I put this antibody on a column, for example, so that way just the DNA pieces that have this red transcription factor get captured, while, for example, this piece that doesn't have the red transcription factor gets washed out. And so now I have all the DNA sequences where this transcription factor binds to, and now basically what I want to do is sequence those DNA pieces to see where that transcription factor binds to. And so for that, I have to reverse the crosslinks. I have to remove the DNA binding proteins from the DNA. So that's sort of the next step. And then basically, I can figure out what are these DNA sequences where this protein, this transcription factor, binds to. So the way to figure it out, one is you can just do qPCR on one region. So you can design primers, let's say on this region, here and here. And you want to see if you get enrichment, if you get a PCR product that's much stronger in your chip versus your control. And the control is usually doing exactly the same thing, but not having an antibody. So if you don't have an antibody, you shouldn't be enriching for a certain sequence. And so with the antibody, you should be enriching for it. So if you do a PCR for this, where you have the antibody, versus one where you don't have any antibody and you're not enriching for these sequences, you also have these sequences in there and so forth, you should see a much stronger PCR band, or measured by qPCR, for those that have the antibody. Is that clear for everybody? Okay, so that's by qPCR. Another way that people used to do this is by chip. You take your DNA and you throw it on an array that covers the genome, and you see what sequences get enriched. But nowadays with sequencing, everybody's just taking all of this, throwing it on the sequencer, and you basically get all the sequences in the genome where this antibody binds to. And that's usually the standard and almost costs as much as doing now a few PCRs. So it's much more widely used, and again, you get a genome-wide sort of view of where this transcription factor binds to. Is that clear? Because I'll talk a lot about ChIP-seq later on. Yeah?
Which step does qPCR happen?
So at the end, so when you have the DNA, you can do a qPCR. So let's say you're interested in this region, let's say region A. So you can design primers that will PCR this region A. And technically, if your chip worked and you enrich for this region, you should get much higher sort of, if you think about a band, you must get a much higher band than if you don't enrich for it, then you don't have an antibody, and you're getting all possible sequences. Is that clear?
So how long would it be the chopped sequence?
How long? Oh, the sequence size. Yeah, so usually it's you design it to be about 250 base pairs, between 250 to 500 base pairs for libraries, for Illumina libraries.
No, not library, but typically you take the DNA and put this on the body, and basically that's what that DNA replicates is something you chopped before.
Yeah, so you chop the DNA before with a sonicator, usually. That's what's routinely used. Or there's enzymes that you can use, but people usually use a sonicator, where so one of the first things when we do a chip in the lab on a certain tissue or a cell is we take the cells and we cross-link them, and then we actually sonicate them in different conditions, and we want to figure out which condition gives us sort of the size that we want, which is on average 250 base pairs.
So how accurately can you measure or estimate
So that's a great question. So with transcription factors, it's not so you'd get about a peak. It's what's called a peak, which we'll show you later on. You get a peak, which is about 250 base pairs to 300 base pairs. I'd say 300 base pair average. So you don't really know where in that 300 base pair it binds. There are other ways, which I won't talk about today, but I'm happy to talk later. That is chip exo, which is a technique that really gives you, it's a way to basically really figure out where it binds. It's a bit more complicated to do, but you can really get a sense of where that transcription factor binding is taking place. But usually people just do standard chip where you get these 300 base pair peak, and you can speculate where it binds there. Another thing you can use is transcription factor binding site programs to see if you're getting, for example, if you know what it binds to, where that would be. But it gives you a speculation that it binds there. It doesn't necessarily tell you exactly where it binds. And then I'll talk a little bit more about histone marks, where there you're really getting gigantic peaks, 2 kb peaks or so forth, which I'll talk in a minute. Any other questions on chip? Okay, so then what antibodies do people use for finding these regulatory elements? And there's quite a lot of different antibodies that people use, too much to talk about. I'll just talk about the main ones today. So one simple one, just like I've showed you before, is you can use an antibody for a transcription factor. And that way you find all the regions in the genome where this transcription factor binds to. And that's pretty standard. The problem with this is that chip really depends on the quality of your antibody. It's probably the number one sort of determining thing on how good your chip works. And so if transcription factors, antibodies sometimes aren't as good. A lot of antibodies have been done for proteins, for other things like Western blots or other things that people use. And sometimes it doesn't necessarily mean that the antibody is going to be good for your chip. Your antibody basically has to pick up your transcription factor when it's binding to the DNA, and the conformation of the protein can be different and so forth. So chip for transcription factors could be sometimes problematic, in particular for the TFs. And if you ever want to do a chip TF, it's worth seeing if somebody's done it before for that transcription factor. And if they've done it and it worked for them, that's probably the antibody that you want to use for starters to see if it works. One example from our lab, we had a protein that we wanted to do TF for, and nobody's done it before. So basically what we've done is we've ordered 4 different antibodies, did the chip-seq, and one of them worked. So a lot of times it's sort of a hit and miss there. And you can do some experiments to see if the antibody works well or not, but with chip-seq getting so easy and cheaper, and in the end, sort of your end result is chip-seq, for us it was just simpler to do 4 chip-seqs and see which one works and which one doesn't. But it's definitely a tough thing in terms of chip is the antibody. So other antibodies that are really widely used, one, because they work really, really well for chip, and 2, they give you sort of a global picture. If we go back here, all this will give us is where that specific TF that we're interested in binds to, which is great if you're interested in that transcription factor. But if you're interested more in a global view, like where are all the enhancers, where are all the insulators, or so forth, there are other antibodies that you can use that are really commonly used for histones. So as you know, histones are basically DNA is wrapped up in histones right here. And there's 4 different histone proteins, and then sort of histone 1 that's sort of like a Ziploc seals up here the DNA. And histones have variants also. So for example, H2A here that makes up this ball has a variant called H2AZ, which can be used there's antibodies that are really good for it. And H2AZ has been shown to be a mark for active regions like promoters, enhancers, and so forth. So that's one example. But probably the most commonly used one is histone tails. So histones have tails, like you can see here. And the histone tails have various modifications on their tails, like acetylation, methylation, ubiquitination, simulation, phosphorylation. And based on the modifications on the tails, that can tell you if some region is, for example, active or silenced. Is it a gene? Is it a promoter? Or is it an insulator? And so forth. So a lot of these marks here on the tails of the histones can be used for chip. And the advantage, as I said, is it gives you a global view. You can find all the enhancers, all the promoters, and not just the ones that your transcription factor binds to. And another advantage is that the antibodies for these tails are really very, very good. And so for chip, it usually works very nicely with them. So this is just an example of the nomenclature for this. So for example, here we have histone H3. and it has this tail right here that you can see here. And you can see here, these are all the different modifications that you can have. You can also have cleavage of this tail, which I won't talk about. But in terms of the nomenclature, and some of these sort of can be activated, some can be repressed, and so forth, based on the sort of where you have the position and what you have on the tail. So in terms of nomenclature, for example, this is position number 3. Number 4. So this is Histone H3. Position number 4, and this is a lysine. So at position number 4 of lysine, so H3K4 lysine, you have a methylation here. And so this would be called H3K4 methylation. If it's right here, for example, this is H3, this is lysine 18, and you have an acetylation here, so this would be called H3K18 acetylation. You can have one methyl group here. You can have 3 methyl group here, so this would be called H3K4 trimethylation. This one would be called H3K4 monomethylation. So this is just to make sure we're all in line with all the nomenclature for this. Is this clear for everybody? Because I'll use this a lot later on. Okay, so that's how people basically call these modifications. And as I said, there are various antibodies for all of these different modifications that can really allow us to find, sort of in a genome-wide manner, a lot of these regulatory elements. And this is a nice slide from this article that I liked that shows all the different modifications for the different histones. And as you can see, yeah.
Sorry, question on the previous slide. How can you tell if they're active, the different categories that repressive or activated marks? Are those clear from the position and the type?
So yeah, so it's been clear for a lot of work from other people that have mapped these and figured them out, some recently, some way back. So people know usually if you have, and we'll talk a little bit about which mark does what, but there's been a lot of work of basically doing a lot of ChIP-seq and seeing where these things fall in the genome. Do they fall on promoters or do they fall on enhancers? And past work on a single basis also. So yeah, and some of them are not as well known actually still today, so I think some of them still be very interesting to look. And some tissues also have unique marks. Olfactory receptors, going back to that, have actually kind of unique silencing marks that other tissues still have not been found to have.
So if someone's looking for something, if someone's finds something new, they would have to do some sort of analysis to figure it out.
Yeah, so the easiest is to do some ChIP, for example, find all the regions in the genome and then align them. And let's say you align them and you see they all fall near introns, just throwing it. So you would think maybe it's a mark for introns or something like that. So yeah, that's probably the easiest way to do nowadays. In the past, it was much more specific. So here you can see sort of a list of all the different modifications. This is each histone, each modification that it has, and sort of what it tells you that it's sort of marking in the genome. And so this is sort of another code that you can think about in terms of the DNA, and one that's, especially for ChIP-seq and regulatory elements, is very useful for people. So what antibodies do we use for each mark that I just showed you, or for each sort of regulatory element? So for genes, for example, if I want to find a gene that's active and transcribed, I can use an antibody for H3, so this is histone 3, lysine 36 trimethylation, so 3 methylation groups on histone 3, lysine 36. So usually transcribed genes would have this mark on the tail, and so if I use an antibody in ChIP for this, I can easily find all the transcribed genes in the genome, similar to RNA-seq, for example. If you all know, I can find, using this, all the genes that are actively being transcribed. So that could be a great mark for that. Clear? All right. So for promoters, there's quite a few different things that we could use. So H3K4. So let's start with this one. So RNA polymerase, it binds to the right near the promoter and starts basically transcribing. And there's an antibody for RNA polymerase that people use to find all the transcribed genes and the active promoters. And so that's a simple one, and that's more of a sort of like a protein, not a histone mark. But in terms of histone marks, what people have seen is that in the promoter, you usually get a lot more of H3K4 trimethylation and less H3K4 monomethylation, so depletion. So if you see sequences where you have a lot of H3K4, so you have 3 methyl groups instead of 1, those are more likely to be promoters. And that's one way to find promoters. Other marks for promoters are H3K9 acylation and H3K27 monomethylation, and there are others also that people use. Going back to enhancers, so enhancers, there's quite a few marks, like these listed here. Going back to this mark where you have a lot of H3K trimethylation versus mono, enhancers are the opposite. You usually get a lot of more mono and less tri. So if you find a sequence where you have a lot more mono, less tri, that's probably an enhancer. If you find a sequence that has more tri, less mono, that's probably a promoter. Probably the one that's most used is H3K27 acetylation. And the reason is that what people have seen is that this mark is actually really good for active enhancers. So enhancers are really active at that time point, while H3K4 mono is more for both poised and active enhancers. So enhancers that are not active but might be active later on, but this one really gives you the active ones. Yeah.
So is there some kind of idea of why there's the opposite behaviors of these H3K4 monomethylation and H3 trimethylation?
Yeah, I don't. Yeah, so this one basically has more methylation, which changes something in the histone in terms of its accessibility, I guess. I'm terrible at this, so I don't know what is sort of the biochemical reason for this. But there is definitely probably a biochemical reason why having two methyl groups makes this sort of more of a promoter and more open, more active. It might be more active, and this needs to be a bit less active or less open. But I'm not.
Promoters and enhancers, do they both kind of affect upregulation of the gene?
Yeah, so they're both activating the gene, though a promoter can be. So this finds active promoters. It doesn't find all promoters. It will find you active promoters. A promoter can also be very silenced, and then you would find probably a very different mark here. And this mark that I just mentioned, H3K27, that's an active mark. That's probably the most widely used now for enhancers. You also see it a lot of times on promoters, and you can use it also for active promoters. So there's a lot of crosstalk between these also. Any other questions on this? Yeah. So the thought is that poise is sort of it's ready, it's on the starting line, but no gun has been sounded yet, and then the active is it's already active and working and so forth. So that's sort of the metaphor for that. And it's been seen that a lot of enhancers are poised. They're usually ready, but something has to activate them to make them active. Other ones, I mentioned H2AZ, and another one that's used a lot for enhancers is P300, which I'll talk about and give you an example for. So P300 is a histone acetyl transferase. So basically what it does is it throws acetyl groups on basically on histones at H, and so that opens up basically the region and makes it more active. And what people have seen that it definitely tends to co-localize with enhancers, both poised and active. And what I mentioned before in terms of P300, and I gave you that example of 85% are to a certain tissue, and 80% the beginning where I showed you that ChIP-seq can be better for finding enhancers versus comparative genomics or comparative genomics and TS. This is from this article from my former lab up here on the Berkeley lab, where they used P300 in ChIP-seq to find enhancers. And what they did here is basically take tissue from the limb for the forebrain and from the midbrain, did ChIP with P300, and they find about 2,500 peaks in the genome. That could be 2,100 peaks in the genome. It could be in the limb, 2,500 so in the forebrain, 561 in the midbrain. And what they showed later is that if, for example, if you take the forebrain peak and check it in the mouse assay, and we'll talk later on about enhancer assays, this is definitely a forebrain enhancer. This one is a midbrain enhancer. And here you can see a nice peak in the limb, and it's definitely a limb enhancer. And they got, as I mentioned, a really good success rate compared to conservation. Again, going back to the first slide that I showed you, So if you look at just conservation, usually what you get is about a 50% success rate of the sequence actually being an enhancer. But if you look in terms of just finding it being a forebrain, it's actually random, right? You're looking at conservation, but you don't know what that sequence is doing. It could be near a brain gene, and you can think that it might be doing forebrain enhancer activity, but you really don't know. While here, the advantage of CHIP is that you're starting with the tissue that you're interested in, right? So if you're starting with a limb and doing CHIP on an antibody that finds you enhancers, you're speculating that you're going to find a lot of limb enhancers. And so that's another advantage of CHIP. And if you look at tissue specificity here in dark blue, you see that 80% of your sequences tend to be enhancers in the limb and not just anywhere else. And so it gives you very good tissue specificity, or here, or so forth. Is that clear for CHIP? Okay, so that's for enhancers. And then, of course, we mentioned silencers. There's a few marks that basically light up silencers. Probably the most used one there is H3K27 trimethylation. So similar to H3K27 acetylation. So if you have acetylated lysine here, you're usually active. If you have 3 methyl groups on that same residue, you're usually silenced. And so that is usually a good mark for this, but there are other marks for silencing. And of course, there's DNA methylation. Just having the DNA be methylated here usually suggests that things are silenced in that region. And then finally, we talked about insulators. In mammals, there's not a lot of them found. The best one is CTCF, which is a transcription factor that binds to insulators, but also other things. And just to show you again the advantage of CTCF, this is work by Bing Reng's group from a while ago, where they did ChIP-seq for CTCF to find insulators. What they showed also is that insulators are pretty common among other tissues. So it looks like enhancers are the main group that sort of changes between tissues and defines tissue specificity. Insulators didn't change much. But going back to the logo plot that I showed you, this was sort of the consensus that they had in the past for CTCF, but now that you get all these tons of binding sites throughout the genome, you can see that you get a much more refined logo plot and a much better understanding of what it binds to. Sorry. So CTCF is a transcription factor that is known to bind to specific sequences and work as an insulator. And so it could be a good mark for insulators. However, it has been shown also to bind to other things. But currently, for mammalians, it's probably the best mark for insulators. And an antibody for it in the ChIP-seq could be used to find insulators. Any other questions on ChIP before we go on? Okay, so another commonly used methods are DNase-seq or attack-seq. And they take advantage of basically open chromatin regions. So as I mentioned, DNA is wrapped up in nucleosomes. But when something is active, for example, if you have here a promoter that's active, this region sort of becomes open and free of nucleosome regions, as you can see here. Or this enhancer right here can also be open, free of nucleosome regions. And so what you can do is take advantage of DNase. So you can use DNase, and DNase will cut very well things that don't have something right here, like nucleosomes that sort of basically fold the DNA and make it harder for the DNase to cut. And so things like this that are active will get cut much easier than things here that are not active. And you can take advantage of that, of finding basically active regions in the genome. And so this is sort of a slide that shows the DNase-seq kind of a pipeline where you isolate cells or nuclei that you're interested in. And as I mentioned, there are things that are open, like here, things that have nucleosomes on it, things that have transcription factors binding to it. And so when you digest with DNase, you get these different fractions of things with nucleosomes, with transcription factors, for example, that are binding, or just open regions. And that you can fractionate by size and actually make a library sequence and find all of the open regions in the genome, suggesting that they could be active. Now, one thing to take in mind, it doesn't mean that they're active in the sense that they're promoters or enhancers. They could be active also. There's binding there, or there's things that go there. That it could be insulators or silencers also. So DNAs-seq basically gives you all the regions in the genome that have some kind of function. You don't know the function of what they're doing. They could be promoters, enhancers, transcribed genes, and so forth. So what really is nice to do is overlay DNAs-seq with your ChIP-seq. The advantage of ChIP-seq, if we go all the way back here, is that you have all these things that tell you what these sequences are doing, right? Silencers should have this. So if you have a DNAs hypersensitive site and you have ChIP-seq for the same tissue that has an H3K27 trimethylation, you would think that that thing is more of a silencer. If you have H3K27 acetylation in the DNAs mark, you'd think that might be more of an active enhancer or so forth. Clear? And so DNAs-seq is sort of was used by only a few, it's a handful of labs. This technique is quite hard to do, and you really have to be good to do it. But recently, there's sort of been a revolution in that sense with a technique called ATAC-seq. So ATAC-seq basically takes advantage of transposomes. So again, if you have regions with closed chromatin and regions with open chromatin, what you can take advantage of is transposomes that can jump and tend to jump into active regions. And what's nice about it also is you can put Illumina primers on these transposomes. And so you have things that jump with Illumina sequencing primers into active regions, and then you just make a library and sequence. So it's probably one of the easiest methods ever. And William Greenleaf, who's one of the people who developed it in Stanford, says, and I'm quoting him, it's so easy that even a PI can do it. And it's really easy. It takes one day, and you get results. And what's nice about it also is that you can do it with very little starting material. And so now even a few months ago, there's an article doing ATAC-seq on single cells. And so it's really a cool technique to use. And we've been doing it quite a lot now in the lab. And it's really, really easy versus DNAs-seq, which really was a complex sort of thing to do. So this is really taken off in a lot of labs, again, due to the ease and the ability to do it on a small number of cells. And so ATAC-seq has been great. So that's sort of how we can find them. Now we'll move a bit more quickly into how far away to look for them. And so there are different ways to look for how far away. So a lot of times, let's say we're interested in this gene right here. And we know, for example, that this gene leads to disease, but we have a lot of families and we have no mutations in the gene that is known to lead to their disease. So we think maybe it's regulatory elements that could be causing this. And as I mentioned, regulatory elements could regulate in cis, so on the same chromosomal position, or they can do in trans from another chromosome. But if we want to look for regulatory elements in cis, there are a few ways that you could do this. One is using synteny blocks, which is, again, you're taking advantage of comparative genomics, which is a bit old, but you can compare, for example, human to mouse, and you can see that this block remained conserved between human and mouse, suggesting that these two regulatory elements probably regulate this gene, and this one probably regulates something else. You can go further in evolution, chicken, frog, and so forth, and generate what's called a synteny block here that suggests that these elements probably regulate this gene, and there was some kind of selective pressure that gave these two together, because if you lose any one of them, this gene now leads to some kind of a phenotype that might be under selection. Clear in terms of synteny blocks? So people have generated a lot of these synteny blocks to give you a sense of how far away you could look for regulatory elements of your gene. So if I'm interested in this gene and I want to find the regulatory elements around them, I probably only search in this block to find regulatory elements that might regulate it in cis on the same chromosome. And this is just an example of a synteny block, which I won't worry you. Other things that people use is just sometimes fixed distance. You have a gene, you go 50 kb away and look for regulatory elements around it. You go 100 kb away, or you go to the next neighboring gene, left neighboring gene. And if you do that, a lot of times we find enhancers and introns of a neighboring gene, so worth going all the way after the left and right neighbor. But nowadays, I mean, the main way we sort of go and look for cis regulatory elements is through topological domains, which I'll talk about a bit later on. But basically, what people have seen is that DNA is divided into these domains that are conserved between tissues and between species. And basically you have this region here where a lot of things interact with one another, but you get much less interaction with this other region here. So if your gene, for example, was in here, I would look for cis regulatory elements only all the way up to here and up to way here, but not past this. So the topological domain sort of can give you how far away to look for your gene of interest for cis regulatory elements. Is this clear in terms of topological domains? Okay. So functional assays to test for their function, and so this is a lot of what we do in the lab. And so we asked for, there was a question before on promoters, and so for promoters, basically it's quite simple. You take a reporter gene like GFP or LAC-Z or so forth, stick in a sequence that you think is a promoter, and you can then stick it into a cell, a mouse, a fish, a chicken, so forth, and see where it turns on. And if it turns on where you think that gene is expressed, you can speculate that that sequence is sufficient to turn on promoter activity. Enhancers, very similar to this. You have a reporter gene, and then you have what's called a minimal promoter. So a minimal promoter is usually like a light switch that doesn't work unless it has an enhancer in front of it. So if you have an enhancer in front of it, that will flip that light switch, and that will turn on the reporter gene. So it's a promoter that technically shouldn't work, but only if it has an enhancer in front of it, that will turn it on. And again, you stick it into something and see if it turns on the reporter gene. One, for example, commonly used minimal promoter in mice is HSP68, a heat shock promoter 68. And so it should only be turned on if you heat shock the animal, which usually doesn't happen. And so that's a great minimal promoter. If you have an enhancer, it turns it on, turns on the reporter gene. Clear? Okay, so how do we test for enhancers or promoters? So as I mentioned, you can take the promoter vector or the enhancer vector, you stick it, for example, into cells, and luciferase is probably the most commonly used reporter gene because it gives you a quantitative measurement, and you have what's called a luminometer that basically measures how much luciferase you're getting from either a promoter or an enhancer. And for example, you have a sequence here that's a promoter versus, and sort of your controls usually having this vector without this sequence. And so you can see, for example, here, this is probably a promoter because you get high expression with the sequence here and no expression without that sequence here, suggesting that that's a promoter. Clear in terms of this? Okay, so the advantage of cell culture is that it's quick, it's easy, it's cheap, and it's quantitative. So with luciferase, you really get a quantitative readout of how strong that enhancer is or how strong that promoter is. The disadvantage is it's not in vivo, it's not the whole organism. So, as cells, especially differentiated cells or established cell lines, are not really, a liver is not really a HepG2 cell and so forth. So it could be quite different. And cultured cells, as I said, tend to lose their tissue characteristics. And there's a lot of variability that we see with this. Sometimes if I make this vector and then I run out of it and then I make another vector, I can get quite some differences sometimes in results and so forth. So for us, we look at it as sort of a plus or minus, but not something in the middle. It can tell you if something's a very strong enhancer or a very strong promoter. But usually in the middle ground, it's a bit grayish sometimes. But that being said, it's a great way to get a sense of something. If it's an enhancer or promoter, as I said, it's cheap, quantitative. We can really scale it up. And in our lab, and I'll talk about it later on, we've been doing technologies where you can test, for example, 100,000 sequences for enhancer activity or 100,000 sequences for promoter activity all in one experiment. And so it's really a great way to do all of these things.
So is there any similarities or differences between the promoters and enhancers?
In terms of the
sequence?
On the sequence level? So there are some differences, though there are some things that are very similar. So the question is yes and no. There are different types of promoters. I didn't go into it in terms of time, but there's promoters with Tata box, which you see more in housekeeping genes. There's Tata-less promoters also. There's a whole sort of like a whole lecture that could be given on different types of promoters. But promoters also there's the core promoter, and then there's the, I'm blanking on the definition. So right next to the core promoter is sequences where you have a lot of transcription factor binding sites, which could be very similar to enhancers. And actually people have, there's a few articles of people have been using promoters to find enhancers. And so based on the transcription factor binding sites of the promoter, can we find enhancers that look similar, and do they regulate each one? So so the short answer is yes and no. Okay, so another widely used assay is Zebrafish. You can take the promoter, the reporter gene, or the enhancer minimal promoter reporter gene, tested in Zebrafish. The advantage of Zebrafish, and we do quite a lot of Zebrafish actually in our lab, is it's quick, it's cheap. You get numerous embryos, so from just one mating, you usually get about 100 embryos. And you can track fish over time. So if you're using a GFP, for example, a reporter gene, you can look and see things over time, and you don't, like in mice, which I'll show you in a minute, you have to take it out of the embryo. So we can follow, for example, an enhancer all the way from development to adult and see how it changes in terms of its expression or so forth. And disadvantage is that it's distant to human. And a lot of times you get mosaicism and you have to generate stable lines, which takes you quite a lot of time. And just like mice, it takes about 3 months for a zebrafish to be mature, and then you can mate it and generate a stable line. So it's great for development and doing quick, high throughput things. It's not great if you really want to go more later on and more high. And again, it's distant to human, but it has a lot of its advantages. Other ways that people use is chicken electroporation. You can take these sequences, electroporate into a chicken, and then see where they light up. Again, advantage is it's quick and cheap. Disadvantage, it's only accessible in the regions that you can actually electroporate in the chicken. Usually electroporation doesn't get into all the cells, so it's expressed in some cells, so you have variable expression. And you have to take out the embryos and look at them, and again, distant to humans. So probably one of the more commonly used ones is mice. And mice, again, you take this sequence, make a transgenic mouse where you stick it into a one-cell embryo. And then in this case, you take out the embryo, this embryonic day 11 and a half. So after 11 days, you take out the embryo, stain it in this case for LAC-Z. And here you see a very strong limb enhancer, suggesting that this sequence of limb enhancer turned on the minimal promoter, turned on the reporter gene. So the advantage here, closer to humans, it's a mammal. DNA usually integrates in one location, and so that gives you advantage. The disadvantage is it's not that high throughput. It takes a lot of time to do these experiments, and they're very expensive. For example, in UCSF to do a transgenic, it cost me now about $3,000. So it's quite a lot of money. And then embryos, need to be taken out for each time point. So you can only look at that time point that you're taking out the embryo versus zebrafish, for example, where you just follow it over time. And so that's also a disadvantage for mice. So all of this depends on basically what you want to do and what you want to get from your experiment and what's the question you're asking. And I think based on that, I think I would choose the system to use based on its advantages and disadvantages. And in terms of mice, there's a big project here in the Berkeley lab, down by my former lab, Eddie Rubin, Len Pennacchio, and Axel Wiesel, where they've characterized a lot of enhancers using mice. This is the URL for this, and you can check out all the sequences they tested. They've tested so far 2,300 sequences, and about half of them are enhancers, and all of these are online, where you can go in and check if you're interested in a certain gene. You can see if they've characterized an enhancer for this. Just to illustrate, though, how not high throughput this took them about 11 years to test about 2,317 sequences. So it takes a lot of time, and if you multiply it times about, let's say, $3,000 for each one, it's quite expensive also. So again, it's not a high throughput way, but it's a great way to really see where things are expressed in a mammalian organism. So switching gears to silencers. So as I mentioned, there are silencers, and there's a few silencers assays out there. They're not that great. They work on the idea that you have a reporter, and in this case, a constitutive promoter, so a promoter that's always on, that's very strong. And if you have something that's a silencer, you would speculate that it will turn off this very strong promoter, and that could lead to turning off the reporter gene expression or reducing it. Another way to look for silencers, just like the other vector that I showed you, enhancer, minimal promoter, reporter gene, but you put a silencer here, and again, does that reduce enhancer activity? So these two things are used usually for silencers. It's harder to see something that gets turned off versus something that gets turned on, and that's one of the reasons sort of enhancers are much more easier to find and more sort of looked at. Insulators, same thing, minimal promoter, reporter gene, enhancer, and the thought is you have an insulator here that will not let your enhancer bind to this minimal promoter and turn it on, and then you're looking for turning off gene expression. Clear so far? Okay, and then of course you can use this in cell culture, mice, so forth, and so on. So we've covered all of this. Now I'm going to quickly go into, since in terms of time, I guess we're running short, regulation in 3D, and then give you one example that sort of ties everything all up from the lab. So one very important thing and one very take-home message is that DNA is not sort of sitting there in the nucleus and so forth, but it has various different regions where it can sit, and there's various different interactions between it. And the interactions have been shown to be very important in terms of gene regulation. And this is a nice picture from a figure where you can see things that interact with one another, both in cis and in trans, and loop to one another. Enhancers can loop to promoters, silencers can loop to promoters, and so forth, and so on. So you really have to think about DNA regulation and gene regulation in sort of a three-dimensional kind of a plot. And there's been quite a few technologies now that take advantage of this actually to find regulatory regions. And so, for example, enhancers are known to bind to promoters. So if you have an enhancer here and a promoter here, in a region where the enhancer is not active, usually you don't have binding. But in a region where it's active, this enhancer loops all the way to the promoter, and you have binding here and a loop. So you can take advantage of that. And there's a technology called 3C. It was the first one that was invented, which means chromosome conformation capture. So you have 3 Cs, chromosome conformation capture. And then later on, derivatives of it are 4C, 5C, and high Cs, sort of whole genome one. And I won't talk about all of them. Again, we're running low on time, but just briefly go through the idea of 3C, which everything else, all of these technologies are based on, just so you understand the theory behind it. So if you have an enhancer that loops to the promoter, you get this loop, and you get now the enhancer right next to the promoter. So you can take advantage of that by way of, just like I mentioned with CHIP, you can use formaldehyde to cross-link the DNA binding proteins to the DNA. So you use that, and now you have the enhancer sitting right next to the promoter due to having these binding proteins. You then cut the DNA, either with restriction enzymes or DNAs or other things. And so now you have these two loose ends that are cut. And then you do a ligation using very dilute conditions, and this is sort of the key step, where only things that are in close proximity to one another will ligate to one another. So, so if your ligation works, you now have basically your enhancer right next to your promoter in terms of a DNA sequence. So then you can reverse across links, and you can do either next generation sequencing or just a qPCR where you have a primer from here and a primer from here. And that should give you a product. While, let's say, this enhancer is 1 megabase away from the promoter, if you do a PCR on where they don't interact and they don't loop, a 1 megabase PCR will not work. It's just too far away, right? It's for a PCR to work. So this PCR will give you something. This one will not give you something. And so that's something measurable that you can quantify and see if you get an interaction between these two. As I said, there's methods where people have developed, like Hi-C that's commonly used, where you can do this in a whole genome sequencing. If you want to read more, this is the first article about it. And another method that's being commonly used is ChiaPET, which is a bit more hard to do, but the advantage of ChiaPET is that you can screen for specific interactions that you want. So if I go back here, this will give me all the interactions in the genome. I'm just showing you the enhancer and the promoter, but there are other interactions in the genome that are happening. With ChiaPET, what you do is you have an extra step that's similar to CHIP, where you use an antibody. So let's say I use an antibody that's specific for promoters, like RNA polymerase, that can pick up promoters. So that way I can get just the promoter interactions in the genome. And when I sequence them, it gives me much better resolution, where I'm sort of selecting just for the interactions that I'm interested, in this case, the promoter interactions. So ChiaPET is basically a chip strung on to a Hi-C. Is that clear for everybody? So the advantage is the resolution. When you sequence Hi-C, you get, now the resolution is much better with improvement of technologies, but usually you get 5 to 10 kb resolution of interactions. So these are really big, and you don't know what's interacting with what. The ChiaPET is sort of a filter to look at the interactions that you're interested in, for example, promoter interactions. So using Hi-C, going back to topological domains, people have seen that there's these domains where things tend to interact a lot between one another, and then there is crosstalk, but not as much between these domains. And then going back to how far away you look for an enhancer, you can use these domains and based on this. And these domains seem to be conserved between different tissues, different cell types, and different mammalian organisms, at least have been shown. And so you can use them, again, in the question of how far away do you want to look for an enhancer for your gene. You can look at where is it, the topological domains, and where are the boundaries for it. Another method to look at interactions is DNA fish, and I'll quickly go through this, and this is just an example of one of this. So here we have a gene called sonic hedgehog, and mutations in this gene in humans in the coding region lead to holoprosencephaly, limb malformations, a lot of bad things. And then 1 million base pairs from sonic hedgehog is the sonic hedgehog limb enhancer that turns this gene on in the limb. If you knock it out in mice, you get these limb truncation phenotype that you can see here, and this is an enhancer, not a gene. And if you sequence patients who have an extra finger polydactyly, they all have mutations in this enhancer. So one group, Shirahishi's group in Japan, actually wanted to show that this enhancer actually interacts with sonic hedgehog and turns it on 1 megabase away. And so one way to do that was DNA fish, and the idea is that you can have a probe here that is red, that targets your promoter of the gene here. And you have another probe here that targets your enhancer that is green. And if you look at the limb, and sonic hedgehog is expressed right here at this portion in the limb, if it interacts, then you have looping basically right here in the limb. And what you can see here is that this is a region where it doesn't interact, and you can see you see the red and the green are not close to one another. But in this region where it does interact, you can see that the red and green are one on top of each other, suggesting that you have looping here to interact. But here, where it doesn't interact, no looping. Clear for this technique? So it's basically taking advantage of chromosomes and sort of cytogenetics, old cytogenetics, to show interactions versus non-interactions. So last part, I'll stop with one example from our lab. This will be just a few minutes to sort of put everything together. And this is work by a very talented postdoc in my lab, Ramon Birnbaum, who now has his own lab in Israel. And he was really interested in this disease called split hand and foot malformation. This is the phenotype that you can see, and we have quite a lot of patients that we've collected in the lab. Split hand and foot is thought to be, it was mapped to these regions, these chromosomal regions, and he was specifically interested in this one, split hand and foot 1, which was mapped to be near DLX 5 and 6. However, a lot of the patients did not have mutations in the gene DLX 5 and 6. The reason people think that DLX 5 and 6 are the sort of the culprit genes is that when you take, both of them are expressed in the limb, and when you knock both of them out in mice, this is what you get, the split hand and foot phenotype. So there's pretty good evidence that knocking out these genes leads to this phenotype, and mutations in these genes lead to these phenotypes. And now there's actually 2 mutations in DLX5 that have been found in patients that lead to to split hand and foot, but most mutations actually have no mutations in these genes. So he really wanted to find the enhancers for DLX5 and 6, and so the way he did this was look at, there's a lot of deletions and translocations in patients. So for example, all of these deletions lead to split hand and foot. All of these did not lead to split hand and foot. There's some inversions and translocations also. And basically using all of them, I won't bore you with this, he found a region where we think the split hand and foot enhancers could be here because of all these clinical studies. And so the next thing that we did is comparative genomics, just like I showed you before, and look for regions that are conserved all the way in evolution that are near DLX5 and 6. So DLX5 and 6 are here, and we're looking at this critical region to find things that could be enhancers that are conserved. For example, this sequence conserved all the way down to fish. And so we found about 29 sequences that are conserved between human and frog, and we started testing them for enhancer activity with the enhancer assay that I just showed you in zebrafish, where we take the sequence that is conserved, throw it in front of a male promoter reporter gene, stick it into the fish, and see does it light up in the limb, in this case in the fin, suggesting that it could be a fin enhancer, a limb enhancer that's altered in that patient. And we also did this in mice, same thing, what I showed you before. So, and this is the region, DLX5 and 6, and you can see we are finding enhancers, but none of them were actually in the limb. So this one was a branchial arch, This one was an ear enhancer, and some patients here have actually hearing loss and so forth. But we weren't finding the limb enhancers for this. So luckily at the time, ChIP-seq data sets came out, and the thing with comparative genomics is that, and one ChIP data set that I showed you is this one for the limb, which we looked at. The thing with comparative genomics is that you usually think that things are conserved, like for example, this exon or this exon will probably be conserved in evolution because it has a protein function, and that's why it's conserved, not because it has a regulatory function. And so you would throw it out of anything that you would think is a regulatory element in general. But ChIP-seq, because it looks with an antibody where you have things that could have enhancer marks, is sort of unbiased. And so when we looked, for example, at this data set that I mentioned before for limb peaks, we found 2 peaks that overlap these 2 exons that are coding for protein, but also have an enhancer mark that suggests that it's an enhancer. So again, if I was using comparative genomics, I'd probably ignore it because the conservation is probably coming from protein. And so, sort of ignoring the obvious, like this sign of this guy that I see a lot when I come out of the BART. And so if we look here, when we tested this in zebrafish, this was actually negative, but this one was a very nice fin enhancer. And when we tested in mice, actually both of them were very nice limb enhancers that are also coding exons of this gene, dynein. And you can see here that they're very similar in terms of the expression, both of them, to DLX5, to this gene that's 900 kb away from this. So using ChIP-seq instead of comparative genomics, we can find these. We could do these enhancer assays. And then the next step, what we wanted to show is that they interact. So if I go back here, do these actually turn on these genes? And so going back to the 2 methods that I mentioned, 3C, we could do 3C and see, have a primer here and a primer here and see do they interact. And when we do that, this is in the limb versus a tissue where there's no interaction, the heart. You can see that if we have a primer from here and a primer from here, we get much higher qPCR results suggesting that they interact. And then going to the DNA fish that I just showed you, we can have a back probe here and a probe here. And if we look at the limb versus the heart where they don't interact, we see much more interaction between this probe and this probe, suggesting that they loop and interact. And that's measurable. You can see how many times you see it in the limb versus how many times you see it in the heart. And you can see that they interact with one another. Another thing we could do is do ChIP, which I also mentioned to you. And so you could do ChIP to see what marks do you get for these things versus these things. And so you can see if I do a ChIP for an enhancer for this region, you can see that it has an enhancer mark in the limb, and this is in the limb tissue. But if I do a ChIP for the transcribed gene, for a gene that's active, this gene is not active in the limb, as you can see here. But this gene, DLX5 and DLX6, both of them are very active in the limb. And so using ChIP, I can show that this sequences in the limb probably are limb enhancers, and these are limb genes. And so we could suggest that that might regulate these genes also. So just to sum up, and that was sort of an example, putting everything together. Hopefully it wasn't too fast for you. And really, if you want to think about gene regulation, this is sort of the metaphor I suggest using, where you have all these interactions in the nucleus and things actually move around, connect, disconnect, and so forth. And with that, I'll shut up and happy for any other questions that you have.